Okay, this is group 9, and we're going to go over the role of environmental variation, tick characteristics, and microbial interactions. I am Nefertiti Mims.
I'm Khadijah Council.
I'm Jacob Fitch.
I am Zaire Langdon.
Okay, so to get into our article that we went over and analyzed, the research from this article gave insight of small-scale ecological variation and micro-to-micro interactions in shaping tick microbial communities and the dynamic of tick-borne disease. Researchers looked into how microbial communities within organisms consist of symbionts, commensals, mutualists, and pathogens that co-occur simultaneously and potentially influence each other in the host. By using the Excitus ricinus tick, they were able to test how this tick species plays as a vector for a range of human, domestic, animal, and wildlife pathogens under specific conditions. And here we have an image. Below is a description that says that the development stages of the Excitus ricinus tick, from left to right and top to bottom, is the adult female or the nymph, adult male or the larva. And using the Excitus ricinus, it is the most common tick species in Europe and is influenced by environmental conditions such as temperature and humidity. Other studies found that the bacterial community composition of the specific tick species have mostly environmental and free living bacteria. Also, what makes this species different from other ticks across habitats, geographical sites, and tick life stages and sexes comes down to the large-scale biotic or abiotic factors, which are biotic factors are living things that exist in the ecosystem, while abiotic factors are living things that influence the ecosystem, and as well as small-scale tick-specific effects on microbial communities, direct biotic interactions, and tick genetics. Going into the next slide, to be accurate in results, the researchers explored the importance of environmental factors, tick characteristics, and direct microbial interactions that influence their structure of bacterial communities in the Excitus ricinus ticks. These results were gathered by using a combination of 16S sequencing and joint species distribution modeling, which we'll talk about later in the materials and methods and results.
Okay, so for materials and methods, 3 locations differing in temperature and elevation were used to sample the ricinus ticks. At each location, 3 levels of elevation were taken into consideration, which totaled in 9 sampling sites. The ticks were sampled in June, July, and August. Those results will be shown in Table 1. Tick DNA isolation and quantification of genetics took place in a laminar flow cabinet. Each tick was washed with sterile water and sterilized with 3% hydrogen peroxide. The ticks were cut in half and the DNA was extracted using a blood and tissue kit. Ticks were later genotyped to quantify individual and population level genetic diversity. During the tick microbiota sequencing phase, common tick endosymbionts were discovered in ricinus ticks. And PCR protocol, which is known as polymerase chain reaction, was used to make copies of the DNA and to amplify it to a large enough amount to study it in detail. During the joint species distribution, JSDM frame was used to examine how environmental variables correlate with pathogens and tick endosymbionts. Next slide. So in this map, we have the location of the sampling sites. You see circles, squares, and triangles, which represent the location, and the colors of those circles, squares, and triangles represent the elevation levels. Next slide. So in this chart, you just see the successfully sequenced ricinus ticks. Females were the most successfully sequenced. Next slide. And this table just shows the classification of OTUs based on the literature search for each taxonomic label, labeling it either pathogenic or endosymbiotic. Next slide.
Okay, this is the results for the Excitus ricinus microbiota composition section. Okay, the bacteria community of 92 isolated richness ticks were sequenced, which resulted in a total of 13,214,477 reads. Hold on. Of those 13 million, only 1,656,287 reads were retained after contingency assembly and quality control. And this was a result of there being application in all of the readings, but they were thrown out if they were shorter than 250 base pairs. There was an actual total of 5,181 OTUs identified. And OTU stands for Operation Taxonomic Unit, and this is a way to categorize bacteria based on their sequence similarity. Of that 5,000, a median number of 1,562 quality control reads were reached. And once that median number of OTUs was verified at 500 reads per one sample, the OTU number went down to 89. And this 89, of course, had a confidence interval of 95% from 78.3 to 98.5 OTUs. There were 6 OTUs found in 90% of the samples, and this was CA, metachloria, and that represented OTU 1. Spigmonosus represented OTUs 6, 2, and 7, and Pseudomonas represented OTU 11, and Delphtia represented OTU 12. Together, they formed 50.2% of all reads. Next slide. Okay, so this is the results of the tick microbiota variants partitioning. And this explained that the most of the variation in tick microbiota is related to the actual tick identification, tick ID rather. For the 100 most common OTUs, tick ID had accounted for over 64.1% of the variation in the model. Then you had spatial temporal random effects such as location, site, and month, and this accounted for, this kind of 23.3% of the model, variation of the model. Then you have 56. Then you have fixed effects such as tick life stage and elevation, which accounted for 12.5% of radiation in the variation in the model. When you group the spatial and temporal random effects with the fixed effects, it changes the overall percentage between the tick ID and the fixed effects. This changes the tick ID to 49.9%, and then the fixed effects to 31.8%, and then the fixed effect tick level accounted for the most, which was 17.5%, and the site level accounted for 14.3%. And what this means is that this explains a large proportion of variation in the occurrence of obligate resident pathogens and symbionts than the occurrence of other, which was potentially non-resident OTUs. Next slide. Okay, the final part of the results is the tick-specific and environmental factors related to the OTU occurrence. So tick sex and elevation of sampling site were two of the variables most important to explaining the presence or absence of tick endosymbionts in human and domestic animal and wildlife pathogens. For example, adult female ticks were less likely to harbor endosymbionts, Spiroplasma, Rickettsia, Lariscula, and Rickettsia spp. Slope and aspect serve as some significant predictors of tick endosymbionts and pathogen occurrence, such as ticks from sites facing northwards had a higher probability of harboring Spiroplasma and B. F-Zilli pathogens. Another thing was that ticks from sites from steeper slopes had a higher chance of harboring Rickettsia sp. Another tick factor, such as tick abundance, was accounted for. The higher tick abundance in the region meant that there was showed that there was a higher chance of these ticks in that high abundance area harboring Rickettsia. And another pathogen, another one, CA nearechia. I'm sorry if I pronounced it incorrectly. And that's all for the results.
Implications and conclusion. In conclusion, the results showed that the JDSM framework can contribute to a better understanding of the factors shaping bacterial communities and natural populations, as well as patterns of co-occurrence among microbes. Further, we can interpret the role of small-scale tick-level characteristics rather than large-scale ecological variation in shaping microbial communities of the eye richness. Our main analyzation was that the shared environmental preferences identified patterns of microbial co-occurrence that are consistent with micro-microbe interactions, which then result in pathogen co-infections with ticks, as well as competition between Stereoplasma and a number of human, domestic, animal, or wildlife pathogens. This opens up new and exciting avenues for the control and management of tick-borne diseases in regions with high human disease incidence. Limitations and sources of error. In this experiment, although the study captured extensive environmental variation with the sampling site spanning across an elevational gradient from 630 to 1,580 mastle, a large number of ecological variables was considered in the models. The power of large-scale ecological factors was weak. Focusing more on a small-scale variable rather than large-scale climatic or environmental factors is usually done when modeling tick-borne pathogen prevalence and would have been a more fruitful approach to advance our understanding of microbial composition of natural populations as well as tick-borne disease dynamics. A limitation in this research was the sampling design. There was an unequal amount of sampling distribution across sites. Ticks were collected all the way to the upper level elevation limit, which caused large variations in environmental variables in the models. And that is it for the conclusion. Acknowledgements. I would like to thank everyone for listening to this presentation. Special thanks to Dr. Fagboden for allowing us to do this presentation and being an amazing teacher.